Welcome back. In part 1, we worked through 5 enzyme kinetics questions covering the Michaelis-Menten equation, the Lineweaver-Burke plot, and all 3 classical inhibition types. If you have not watched part 1, I recommend starting there. In this video, we go deeper. 5 more questions. Allosteric regulation. Suicide inhibitors as cancer drugs. Turnover number calculations. And a clinical question about a real drug used to treat leukemia. Question 1. An enzyme displays a Hill coefficient of 2.8 and shows a sigmoidal substrate velocity curve. What does this indicate? Option A. Michaelis-Menten kinetics with an unusually high Km. Option B. Positive cooperativity. Substrate binding at one site increases affinity at others. Option C. Negative cooperativity. Affinity decreases with successive binding events. Option D. Uncompetitive inhibition by the substrate itself. The core concept. The Hill coefficient, N, is the central indicator of cooperativity. When N equals 1, the enzyme follows standard Michaelis-Menten kinetics. A hyperbolic substrate velocity curve. When N is greater than 1, the enzyme shows positive cooperativity. Binding of the first substrate molecule increases the affinity of remaining subunits for substrate. This produces the sigmoidal curve. A Hill coefficient of 2.8 tells you 2 things simultaneously. The enzyme has multiple subunits, and they communicate with each other through conformational changes. Hemoglobin, with an N of approximately 2.8, is the canonical example. The answer is B. The step-by-step reasoning. Step 1. The question gives you a Hill coefficient of 2.8 and a sigmoidal curve. Both together eliminate option A immediately. Michaelis-Menten kinetics produces a hyperbolic curve, not sigmoidal, and has a Hill coefficient of exactly 1. Step 2. Eliminate option C. Negative cooperativity produces a flatter, more hyperbolic curve, not a steeper sigmoidal one. Step 3. Eliminate option D. Substrate inhibition produces a curve that rises then falls at high substrate concentrations, not the classic sigmoidal shape, and not associated with a Hill coefficient above 1. What remains is B. Positive cooperativity. Hill coefficient greater than 1. Sigmoidal curve. These 3 features are inseparable. Now the distractor analysis. Option A. High Km exploits a common confusion. Students sometimes see a sigmoidal curve and assume the enzyme simply has poor affinity for substrate, expressed as high Km. But Km is defined within the Michaelis-Menten framework, which does not apply to cooperative enzymes. The Hill equation replaces it with K0.5. The substrate concentration at half maximal velocity. Option C. Negative cooperativity is a trap for students who know cooperativity can be positive or negative but are unsure of the direction. Negative cooperativity reduces affinity with successive binding events. The curve becomes flatter, not sigmoidal. Option D. Substrate inhibition produces a curve that rises then falls at high substrate concentrations, not the classic sigmoidal shape, and not associated with a Hill coefficient above 1. Let me draw your attention to something in how that explanation was structured. We did not start with the formula for the Hill equation. We started with what the Hill coefficient physically means. Communication between enzyme subunits through conformational change. That physical understanding immediately told us which options were possible and which were not. On DBT JRF Academy. This is called mechanistic elimination. You do not eliminate wrong options by guessing. You eliminate them because you understand the mechanism well enough to know they cannot be true. That skill is what separates Section B preparation from Section C preparation in CSIR NET. A brief note on why enzyme regulation is the most tested topic across all 4 exams. Allosteric regulation appears in every life sciences entrance examination because it is the primary mechanism by which metabolic pathways are controlled in living cells. Suicide inhibition appears in cancer pharmacology questions in CSIR, DBT, and ICMR. Turnover number and catalytic efficiency appear in every quantitative reasoning section. And competitive inhibition of kinases appears in ICMR clinical biochemistry questions regularly. 1 Mechanistic framework. 4 different examination formats. Question 2. 5-fluorouracil acts as a suicide inhibitor of thymidylate synthase, which is required for DNA synthesis. Which statement correctly describes this class of inhibitor? Option A. A reversible competitive inhibitor with unusually high affinity for the active site. Option B. An inhibitor that requires the enzyme's own catalytic mechanism to become irreversibly active. Option C. A non-competitive inhibitor that permanently reduces Vmax. Option D. An allosteric inhibitor that shifts the sigmoidal curve rightward. Suicide inhibitors are one of the most elegant concepts in enzymology, and one of the most clinically important. The mechanism works as follows. The drug enters the active site looking like a normal substrate. The enzyme begins to catalyze it through its own mechanism. Partway through catalysis, the drug is transformed into a highly reactive intermediate inside the active site. That intermediate then forms a covalent bond with the enzyme, permanently inactivating it. The enzyme has, in effect, catalyzed its own destruction. 5-fluorouracil works exactly this way against thymidylate synthase. The answer is B. Note: suicide inhibitors are irreversible. This immediately eliminates options A and D. Option C is eliminated because non-competitive inhibitors do not require the catalytic mechanism to activate them. Question 3. An enzyme has a Vmax of 120 micromoles per minute and a total enzyme concentration of 0.2 micromoles per liter. What is its turnover number? Kcat. In units of per second. Option A. 600 per second. Option B. 10 per second. Option C. 24 per second. Option D. 0.0028 per second. Step 1. The definition. Kcat equals Vmax divided by the total enzyme concentration. Step 2. Calculate. 120 divided by 0.2 equals 600 per minute. Step 3. Unit conversion. 600 per minute divided by 60 seconds per minute equals 10 per second. The answer is B. Option A. 600 is the most common wrong answer. Students forget to convert minutes to seconds. Always check your units. Question 4. The same enzyme has a Kcat of 10 per second and a Km of 0.1 millimolar. What is its catalytic efficiency? Catalytic efficiency equals Kcat divided by Km. Substituting. 10 divided by 0.1 equals 100 per second per millimolar. That equals 100,000 per second per molar. Or 10 to the power 5 m-1 s-1. The answer is A. For context, catalytic perfection is defined as 10 to the power 8 to 10 to the power 9 m-1 s-1. The diffusion limit. This enzyme, at 10 to the power 5, is competent but not diffusion-limited. Enzymes like carbonic anhydrase and catalase operate near diffusion-limited perfection. Question 5. And our clinical closer. Imatinib, sold as Gleevec, transformed the treatment of chronic myeloid leukemia by targeting the BCR-ABL tyrosine kinase. It acts as a competitive inhibitor of BCR-ABL with respect to ATP. What is the predicted kinetic consequence on BCR-ABL activity in the presence of imatinib? Option A. Apparent Km for ATP decreases. Vmax unchanged. Option B. Apparent Km for ATP increases. Vmax unchanged. Option C. Km unchanged. Vmax decreases proportionally to drug concentration. Option D. Both Km and Vmax decrease. This is competitive inhibition. The same mechanism as Part 1. Question 3. Now applied in a clinical context. Imatinib competes with ATP for the kinase active site. Apparent Km for ATP increases. More ATP is required to displace the drug. Vmax is unchanged because at saturating ATP concentrations, the drug is competed out. The answer is B. 5 questions. 2 videos. You now have a complete conceptual framework covering enzyme kinetics from the Michaelis-Menten equation all the way to the clinical pharmacology of cancer drugs. You understand why Km changes in competitive inhibition and not in non-competitive inhibition. You understand what a Hill coefficient above 1 physically means. You understand why suicide inhibitors are irreversible. You understand how to calculate and interpret Kcat and catalytic efficiency. And you understand how imatinib works at the mechanistic level. These are not isolated facts. They are one connected framework. If you found this series useful, please subscribe and share it with one fellow student who is also preparing. JEC JRF Academy links are in the description.